Effect of different concentration of salt or sugar on animal cells. Take 3 slides such that this is slide A, B, and C. Take a drop of blood. Put 1 drop of blood on each slide with the help of a sterilized needle at the tip of finger. Wait for about 5 minutes. After taking a drop of blood on each slide, we take sugar solution A, B, and C. The first one is hypo, the second one is iso, and third one is hyper. Add solution A on slide A. Put some drops of blood on slide A. Spread the blood with the help of clean slide. Now observe the slide under microscope. Now observe the result. In slide A, the RBCs have become swollen due to hemolysis. In slide B, the RBCs maintain their normal oval, biconcave shapes. In slide C, the RBCs have shrunken due to exosmosis.